Hello and welcome to Nikolai's genetics lessons, and today's problem is in the cross of these two genotypes, what is the probability of producing genotype that is going to be homozygous dominant for all three genes. Just looking at this problem I can instantly tell the answer, and I want to teach you also be able to tell instantly and solve such problems very fast. But I'm going to explain you step by step what we are going to do. So we are given two genotypes of the parents, and this is parent 1, this is parent 2. So let me write down these two genotypes one more time. So both parents would be heterozygous for both for three genes. We have three genes here: gene A, gene B, and gene C. And what is the probability that the progeny would belong to this genotype that is going to be homozygous dominant for all three genes. And this is going to be gene A, this is going to be gene B, and this is going to be gene C in, as you see, two parents. So this is going to be parent 1, and this is going to be parent 2. And in the progeny that can of course be of the different genotypes, so this is going to be F1 generation, we are looking for the probability of producing this genotype. And I want you to pay attention that we do different calculations when we looking for phenotype. So in simple Mendelian genetics, two different genotypes, which is heterozygous and homozygous dominant, would produce the same phenotype, but this is going to be different genotypes. So a few minutes ago I uploaded video where we were looking for phenotype probability, but this time in this video we are looking for genotype probability. So because here we have independent assortment, nothing is said that these genes reside on the same chromosome, otherwise these genes would be linked and it would be reflected in our problem that these genes are linked. But because it is not said, that we have to consider that these three genes are on the different chromosomes and would assort independently. That means that we can take each allelic pair and build Punnett square for each allelic pair separately. So parent 1 is heterozygous, parent 2 is also heterozygous for the gene A, and when we build a Punnett square we can see probability for this couple to produce genotype that is homozygous dominant. And as you see, such a probability would be 1 out of 4. So 1 out of 4 would be probability that progeny would be homozygous dominant. For the second allelic pair, both parents also heterozygous, so it's not necessary for me to make another Punnett square. As you understand, we are going to get the same probability, that is going to be 1/4. So 1/4 probability that progeny also would be homozygous dominant for the gene B. And both parents are heterozygous for the gene C. Once again, if we would put these genotypes as parent 1 and parent 2, so this is parent 1 and this is parent 2, we are going to see the same probability, which would be 1/4 for the homozygous dominant genotype. And because this is all independent probabilities, we have to use product rule. And in other words, when we use a product rule, when the question is what is the probability that first allelic pair would be homozygous dominant and second allelic pair would be homozygous dominant and third allelic pair would be also homozygous dominant. When we can connect uh, two or more independent probabilities with the word and, we use a product rule. So 1/4 multiplied by 1/4 is going to be 1/16, multiplied by 1/4 going to be 1 out of 64. This is going to be probability that progeny would be homozygous dominant for all three genes. So as you see, this is answer D. But what if the question would be, what is the probability that the progeny would be of the same phenotype as parent 1 or parent 2? So how this would affect our calculations? All these three, as you see, genotypes would produce the same phenotype. So heterozygous would be the same phenotype as homozygous dominant. So our calculations in this case would be 3/4 multiplied by 3/4 and multiplied by 3/4. So 3 out of 4 would meet our requirements to have the same phenotype as parent 1 for each gene. So as you see, this is very important to pay attention whether we are looking for particular genotype or if we are looking for phenotype. This would affect our calculations. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates and see you in the next video. Goodbye.